Hello all, welcome back to the BioRocks channel. In this video today, I'll be introducing with the different types of DNA sequences in eukaryotes. Okay, how are, see, the thing that you're seeing presently now on the screen are all the nitrogen bases, right? The adenines, the guanines, the thymines, the cytosines. These are all the different sorts of your nitrogen bases, and how these nitrogen bases are being arranged is what I'm talking about. I'm talking about these DNA sequences. How are these nitrogen bases being arranged? So if you're going to look at the DNA sequences, children, the DNA sequences, these may be arranged in two ways. One is you may find some unique DNA sequences which are not repeating, which are not repeating for millions of times. These may be having only few hundreds of copies in the genome, but there might be some rearrangement of the nitrogen base pairs in them, due to which they are not identical to each other, but yet they are going to be the unique DNA sequences. And the second one that you're going to have is you may have these nitrogen base pairs repeating themselves very very commonly, and these will be called as a repetitive DNA sequences. Okay, what are unique sequences are nothing but these are existing as, they are going to exist as the single sequences. These may have only a few copies in the genome, and these are the ones which are encoding your functional proteins. Okay, these are encoding your functional proteins. Got it? But we have another sequences also where you can see that these are continuously repetitive. For example, if I'm going to show you this slide, you can see that these nitrogen bases that you can see that the AG, these are repeating AG. This particular sequence is highly repetitive. So if these sequences are repeating, then we are going to call them as a repetitive DNA sequences. Okay, children, have you got it? And another important point to note here is sometimes in the unique sequences, we may even have some gene families also. Gene family. Okay, what is gene family? Gene family is nothing but sometimes what happens is these existing unique genes, what they are going to do is they are going to be duplicated. Okay, these are going to be duplicated and these are going to be rearranged. These are going to be rearranged. If these are duplicated, a copy of it is being produced. And if they are rearranged, what is happening? They are becoming different to that of from the copy from where they have copied, right? For example, if suppose I'm going to have AG, okay, this is being copied, AG, but here there is rearrangement of these nitrogen bases. Now what has happened is, already I have told you that they are being copied, but there is a rearrangement. So you can see that there are these particular groups are being called as a gene family, we are going to call. Got it? So first of all, what has happened is there is a copy of a gene which has got duplicated. And after duplication, what has happened is they have got rearranged. So now what has happened is from that of the parental gene, this particular gene which is being produced is a bit different because of the rearrangement of these bases, right? So this family is being called as a gene family. For example, if you're going to speak about the gene family of all your antibodies, that is nothing but immunoglobulins, we have different sorts of these gene families. For the cytokines, we have different gene family, and for the chemokines, we are going to have a different gene families. I hope you're understanding. We are just trying to concentrate on how are these nitrogen bases are being arranged. What are those DNA sequences? Either they can be unique DNA sequences which will be encoding for your functional proteins, or you may have some gene families which have got duplicated from an existing gene, rearranged, but they are similar, but they are not identical to each other. They are similar because there are some few of these nitrogen bases which are very much same. For instance, if you can see that in this case that I have written AG is very similar to this, but they are not identical. Why are they not identical, children? Because here this AC has got rearranged. So such families are being called as gene families. And you can see that during the course of evolution, we could see that there are different sorts of gene families that have got evolved. Okay, that is about the unique sequences that we can talk about. So when I spoke about the unique sequences, now it's a duty for me to introduce you with this chromosome. For example, if this is a chromosome structure, children, then the one that you're seeing here is going to be the centromere, and the ones that you can see towards these ends here and here are going to be the telomeres. If I'm going to project this chromatid arm, and if I'm going to project it, now you can see that this is going to show the whole structure in this way. This is the DNA that I am taking up from the chromatid arm of this particular chromosome. So you can see that you are having many gene clusters here, right? These are all the gene clusters that you can see in this particular, what you call is a DNA of this particular chromatid arm of your chromosome. Now, children, if you're going to once isolate one of this particular gene and check out, this particular gene is going to comprise of a most important sequence, which is being called as, this area is being called as a regulatory sequence. And to that off, you can even see the presence of these dark structures here. These are being called as exons. And these exons are actually responsible for what, children? These are responsible for the encoding of proteins, right? These are the exons which are going to code for the proteins. And these exons are interrupted by whom? Who are they? Here you can see this white space, this which is interrupting these exons is being called as what? This is being called as your introns. These are your introns. Once this particular gene is going to be, so that is, I'm speaking about an average gene. This is a particular gene that you can see in a DNA sequence, which is having a regulatory sequence, some of the exons, some of the introns. So once the transcription is going to be done, let me show you. So once if the transcription, after the transcription and after the processing of the hRNA, what are you going to be getting? You are going to get this particular mRNA, which will be of only few thousand bases length. Okay, so you could see that the DNA having the GNA having these regulatory regions, the exons and the introns. Once they are transcribed and they produce a hRNA, and the hRNA is going to process, you get your mRNA, which is of a few thousand base pairs of length. And remember, children, these are these exons which are going to encode for the proteins. Okay, so I was talking about the DNA sequences today, and I spoke about the unique DNA sequences which were encoding for the functional proteins. Sometimes you may even have the gene families also. Okay, that we have seen. Now coming on to repetitive DNA, children. What is repetitive DNA? I have already told you that these particular sequences are going to repeat one. That is, you can see that AG getting repeated all the time. And you can see that this sequence is of how much length is AG. Okay, this is again repeating AG. Okay, it's getting repeated. So these repetitive sequences, we are going to actually call them either as a tandem repeats or interspersed, based on how are they present and where are they present. Okay, children, if the repetitive DNA sequences at the nitrogen bases are very repetitive, and if they are arranged just one after the other as clusters, then you should call them as tandem repeats. In unique sequences, there is no repetition, but in the DNA sequences like these, where you can see that the sequences are every time getting repeated in the same order, so we are calling them as repetitive DNA sequences. If these repetitive DNA sequences are present one after the other, we should call them as what? The tandem repeats. But if your repetitive DNA sequences are dispersed throughout your genome, then you should call these as interspersed DNA sequences. Okay, interspersed DNA sequences, or we can even call them as interspersed elements also we can call them as. Okay, Nachan? So remember that your repetitive DNA, as I told you, let me write it here. The repetitive DNA sequences is of two different types. One is going to be moderately repetitive and the other is going to be highly repetitive. What is moderately repetitive? Moderately repetitive is nothing but, remember that these are going to exist only in a length of 150 to 300 base pairs only. And these tandem repeats are going to be the clusters, and the moderately examples are going to be your tandem repeats, number one. And the second examples that you can see is going to be the interspersed elements that we can see. And these tandem repeats are going to be present. Examples that I can speak about here is going to be the tRNA as well as a ribosomal RNA are the examples for these tandem repeats. Okay, children. Coming on to the interspersed, which are dispersed here and there throughout your genome, these are going to be actually the ones which are transposable elements. These are the actual genes which are going to be called as jumping genes, which will be either copied and pasted to the another section of the genome, or this particular gene will be cut from the area and will be pasted to another section of your genome. That's the reason we call these transposable elements as what we are going to call them as jumping genes. Either they will be copied and pasted or cut and pasted. Okay, and coming on to this particular interspersed ones, interspersed are again divided into the short interspersed elements, which are called as sign, and we have long interspersed elements which are being called as a line. So what is this? This is a short interspersed element. For this, the best example that we can speak is about the ALU sequences. So the ALU sequences are going to be present more than 1 million times in our human genome, and they make out around 11% of the human genome, children. And these are the actual transposons. These are the actual transposons which are going to make multiple copies throughout the human genome. But luckily, these ALU sequence transposons are inactivated throughout the evolution. Okay, these are inactivated throughout the evolution. If not, it would have been produced more multiple copies of these DNA repetitive sequences. So it does not transpose at all. Which are those? The ALU sequences, which are the short interspersed elements. Now, talking about these lines, the long interspersed elements, children, these are structurally very much similar to that of your retroviruses, and these are actually the retrotransposons. These are the retrotransposons. What are retrotransposons? These are those DNA elements which are going to propagate through the RNA mediated transposition. Have you got it, children? How will it be done? Is nothing but DNA will be copied into RNA, and RNA will be back. It is going to be reverse transcripted to the DNA. Okay, so that is going to be the retrotransposons. Okay, retrotransposons. So those are going to be the only long interspersed elements only. So these are the different sorts of the repetitive ones which are moderately. These are the tandems which may be present at one particular area only clustered, but they're very much repetitive DNA, not coding for any protein, as well as they may be sometimes dispersed throughout the genome, not coding for any sort of like protein. And especially, children, you can remember that if we are going to speak about the highly repetitive sequences, the highly repetitive sequences are going to be mostly very less than the 10 base pair long. They are going to be very short in the length, and these are going to be existing in millions of copies throughout our genome. Okay, throughout our genome, and these are mostly present in our centromere, as we have seen in the last video, as well as in the telomere section. You can find these highly repetitive copies of the DNA in your centromeres as well as telomeres, and they are existing as microsatellites. Why do we call them as microsatellites, children? Because when you are going to subject the DNA for the separation by the high speed and the density gradations, then you can see that the tandems are going to be very different from the other sequences, okay? And these repetitive units, what they do is they separate as same as a satellite. So that's the reason we are going to call them as a microsatellites, okay? So when these repeat, see, I'm repeating again, once again, remember why do we call them as microsatellites is when you are going to actually perform the DNA separation techniques by high speed and the high density gradations, what happens is these repetitive units are going to be very different from the other DNA, unique DNA sequences. So what they do is before they decompose, or that is before these are going to actually separate away, these are going to separate in a form of a satellite, okay? They're going to move out as a satellite bodies. Why? Because of their composition. What is their composition? Is nothing but the repetitive base pair units. So we are going to call these as microsatellites, a highly repetitive units. We are going to call them as microsatellites. I hope, children, you have understood that we have gone through the DNA sequences, how they are being arranged in eukaryotic cell. Either they may be unique, they may be having only few copies of these unique sequences, or they may be present as gene families, which are encoding for the functional proteins, or sometimes they may be present as repetitive DNAs, either in form of tandem repeats or interspersed repeats, or sometimes they may be even present as highly repetitive in millions of copies in the telomeres and centromeres also. And lastly, coming on to the differences between the unique and highly repetitive sequences, children, let me highlight these points. So now coming on to, I told you that these are going to either occur once or only in hundreds of copies, but these are going to be in millions of copies. And these are going to be long base sequences. These are very short sequences, sometimes very less than 10 base pairs also, as we have seen. These are going to have the active genes which are coding for the proteins, but they may not, okay? They are going to be translated to the active proteins, but these may not, or sometimes they are ever translated. These may not code for any proteins at all, because mostly these are going to be the sections which are introns, and these are the ones which are going to contain exons, okay? And remember, children, you can see that there is a very little difference between the unique sequences in the different individuals. But the most important highlight point here to remember is these ones which are being called as highly repetitive tandems which are being present are varying a lot in the individual. So why can't we use this particular thing as a prime thing to use in the DNA fingerprinting then, right? So in the forensic medicine and in the DNA fingerprinting, our DNA profiling, since we are going to see that the highly repetitive sequences are very different in the two different individuals, we can use them as a prime markers for the DNA profiling, okay? And these are going to have the highly repetitive DNAs here are going to have mostly introns, maybe very, very repetitive. Okay, introns are very repetitive. These are not coding for any proteins, and these are the small portion of the genome, the unique sequences, while you can see that the higher proportions of the genome are occupied by these highly repetitive sequences. Okay, children, I hope you all have understood about the different sorts of the DNA sequences in the eukaryotes. I hope you all have understood, and if you have liked this video and if this has helped you in understanding the DNA sequences in the short, and so please do try to subscribe my channel and try to like this video, and try to please comment in the comment section how you liked it and how you have this video helped it. Please do comment in the comment section. Thank you all. Thank you.